Yeah, well, we're going to be recording this meeting, as you heard, but yeah, I mean, it's pretty straightforward. Dan will be giving his presentation. I'd like, generally I would ask people to hold their questions till the end. And then afterwards, we'll have a question period. I think we can follow our usual protocol where we invite graduate students to ask questions first, and then faculty members, particularly those of us who are not on the committee, since we're going to have our crack at it tomorrow. So we would invite any outside comments. Other than that, Adrian will be introducing Dan, and I think that's about it.
Okay, great. Thanks, Dietz. So it's my great pleasure to introduce Dan Coppeto. I just want to say this is a huge milestone, of course, for him to be defending his dissertation. And I'm just sad that he won't have the benefit of, the in-person celebrations that usually go along with that. If any of you are interested in helping out on a gift for Dan, we're putting some things together, and so feel free to get in touch with me. For those of you who don't know me, my name's Adrian Yagi, and I was an assistant professor at the anthropology department at Emory for 3 years, and I became the chair of Dan's committee in, I think, January 2017. Mm-hmm. So it was sort of later in Dan's graduate career, and so I also wanted to acknowledge the contributions that other people had made in advising Dan, especially Jim, who was his initial advisor, Paul Hooper, who acted as Dan's chair for, I think, about 1 year, mm-hmm. And then also Dietz, who has filled the formal role of chair in the past year. On top of that, Dan was lucky to have an outstanding committee with Todd and Larry, who both provided resources and expertise that we wouldn't have had within the anthropology department. So thanks very much to all of you for those contributions. Now, I think it's fair to say that Dan had one of the most comprehensive and ambitious dissertation projects ever. So there are lots of people who are interested in the kind of neurobiological basis of behavior. Many people who study oxytocin, some also study vasopressin. In primates, people had only just begun to look at oxytocin using different approaches, comparing oxytocin levels in relation to behavior, comparing different species with different behavioral styles with regard to their oxytocin levels, or in some rare cases, looking at the distribution of oxytocin receptors in the brain. Now, Dan did all of those things, which allowed him to get a much more complete picture of how these neuroendocrine systems actually relate to behavior. Because if it's not the hormone levels, then it must be the receptors, or vice versa. And unlike most people, Dan was actually able to look at all of this in his own research. Another unusual thing is that many people just work with one type of biospecimen for studying hormone levels. So they might have blood samples, or in some rare cases cerebrospinal fluid if they work in a captive setting. If you're in the field, you mostly collect urine samples because they're less invasive. But again, Dan collected all of these different specimen types, which also provided him the rare opportunity to look at how the peripheral and central levels of oxytocin compare and how they relate to different behaviors. So I think in short, it's his work has a huge potential to make contributions to understanding primate behavioral biology and by extension also human behavioral biology. And I think he has some very important publications in the pipeline that will be really nice to see published. Another thing I wanted to just highlight is that Dan also wasn't shy to get into new things like statistical methods, even in the later stages of his project. So he actually voluntarily attended my Anthrostats course that I taught a few years ago, where I forced the students to learn Bayesian statistics. And Dan really took the opportunity to get into R coding and develop a deeper understanding of statistics. And as you'll see in his presentation, he ended up using really state-of-the-art Bayesian models to analyze his data. And finally, if you've ever been cornered by Dan at a cocktail party, you know that he really loves his baboons. And I think many of us will fondly remember the stories of Sergio and the rest of his subjects. And I'm sure he'll treat us to some of those today if we give him a chance. So without further ado, I'm really looking forward to hearing Dan's presentation, and please join me in welcoming him to this virtual stage. The floor is yours, Dan.
Thanks. Yeah, it's the silent applause. Thank you so much, Adrian. That was such a wonderful, sweet, and exaggerated view of me so far. But thank you. Yeah. So hello, everybody. Today, I wanted to talk about my dissertation project, which I have totally not bombastically titled "Assaying the Blood of the Sacred Baboon: Oxytocin, Vasopressin, and the Behavior of the Baboon Subspecies." So we can either do this, or if you prefer, we can also just, take the next hour and a half and just chat about the election. I mean, it's one or the other. I'm sure everybody would love both of those options. But yes, so let me go into this. Now, when I did my dissertation prospectus many years ago, I started with this quote from Charles Darwin. "He who understands baboons would do more towards metaphysics than Locke." And I did it as a bit of a tongue-in-cheek kind of joke, just kidding around saying, well, if Darwin thought working with baboons is great, then it's got to be great. So that is an automatic justification for anything I want to do with baboons. But I also have to admit that, through the course of this dissertation, I have seen in many ways just how useful baboons are to understanding the human condition, which is really the goal of our anthropological field. However, I also have to admit that as a nice bookend to my dissertation, I found another quote that I think would also convince you all that baboons are very relevant to understanding human behavior. This is a quote from Eugene N. Murray, who wrote the book My Friends the Baboons. And his quote was, "The life of the baboon is in fact one continual nightmare of anxiety." And I think everybody can see how much this applies to our own lives. See, for example, all I have to do is change baboon and put in person living in 2020. And this is, I think, very obvious, the connections between us and baboons. But moving on, so my motivation for even undertaking this dissertation was really the result of two observations. First, that came to me was I was always really interested in baboons and how they had such completely different social systems, societies, and especially mating structures related to all these behaviors of affiliation, aggression, and bonding. Now, one of the benefits of being here at Emory University is that we do a lot of research on oxytocin and vasopressin. So from that, I know that these are essential hormones to just those sorts of behaviors and to mediating the differences in social systems between species in these behaviors. Those are my two observations. So my primary question became, how are these baboon species typical behaviors being mediated by these, or being potentially mediated by these different neurohormones? So my primary aims of the study when I started were, one, to do a between species comparison, or between subspecies, to compare the baseline hormone levels between the two subspecies. Then also to do within subspecies, because I was not only interested in why they're so different from each other, but even within, how do these hormones relate to individual temperament or individual social behavior? And lastly, I also undertook a neurobiological comparison where I wanted to look at the differences in the distribution of the receptors for these neurohormones in the brain. Now today, this is already going to be a long one, but I'm only going to focus on the first two. They're the most complete and the most that I can talk about. So I'm going to go through those ones today. So what I'm going to talk about first is basically just say, okay, what are the baboons? Why do I think they're so important and so useful? Then, well, how do oxytocin and vasopressin relate to the baboon question? What can they contribute to it? My hypotheses and my aims that I set out to study, the methods that I used, and then finally, I'll talk about the two studies. The first study being a between subspecies comparison, and the second study being a between individual comparison of social behavior. And then I'll briefly just talk about some conclusions and the future directions that I would like to take this sort of research. So baboon social life. Well, obviously from this picture, we can tell baboons are adorable, but what else can we know from them? How can they help us understand more about humans? Well, baboons are found throughout Africa. The two subspecies that I'm looking at are the Anubis or the olive baboon, which is found here mostly in equatorial Africa, and then also the Hamadryas baboon, which is located here more in the desert regions, hence its alternate name being the desert baboon or the sacred baboon. And in this orange area right there where the two ranges actually meet, that's actually an active hybrid zone where hybrid groups form very naturally. And these hybrid offspring actually have their own hybrid social systems, which incorporate elements of both subspecies. But what exactly are these unique social systems that I'm referring to? Well, for the Anubis baboon, those of you who work with rhesus macaques may realize it's very typical of a standard old world monkey. They live in large multi-male, multi-female troops, and they have a promiscuous mating system, which means that there are no kind of long-term bonded mating relationships. Rather, during a period of female's estrus, she will mate with as many males as possible, and males will mate with whatever females are in estrus at that time. The only real kind of relationship that you see is a consortship, which is essentially a kind of short-term attempt by the male to monopolize female mating during her estrus period. Now, the Anubis baboons are also female phylopatric, meaning that females stay where they're born. This has the consequence of really putting more value onto maintaining your female-female, especially kin relationship. So the female-female bond is the most important for an Anubis baboon. However, that does not mean that there are no intersexual bonds. In fact, baboons have a very interesting relationship called friendship, where it's a non-sexual relationship between a male and a female that's characterized by preferential proximity maintenance and preferential grooming relationships, like these two individuals here, one of whom who's currently scratching his scrotum. But yes, and so it's typically marked by that sort of affiliative behavior. Okay, but what about the Hamadryas baboon? What makes it such a useful comparative model? Well, these are actually some of my own Hamadryas baboons. That's Prince and his unit, which I jokingly referred to as the Revolution. I named all the males in that one after musicians. And so they actually have a multi-level society where what you see is that it goes from being the larger sized troops down to bands and clans, and then finally down to the basic one male unit. Now, this is a male phylopatric community, meaning that the males stay where they're born. And what the consequence of that meaning that, that a lot of these subgroups are made of related males and how they'll interact with each other. However, the strongest bond in these societies is not the bond between males, but rather it's the cross bond. It's the bond between a male and a female. It's actually been referred to quite commonly as a pair bond in this one male unit. Now, one male units or harems, typically in the wild are about 1 to 9 females per one male unit, but the most common number really is to just have one female as part of a unit, just be a male-female dyad. And the important thing about this is that the male sticks with the female despite whatever her reproductive state is. So if she's lactating, if she's pregnant, if she's not in estrus, if she's in estrus, he stays with her during that whole period. And people have even asked, well, maybe are the females just as promiscuous as an Anubis female? And I would actually argue no. I would say that they're not. Based on my observations in captivity, where you have these skewed sex ratios, where you could have one male units or harem sizes up to 18 females to a single male, there's no way he can mate herd all those females. There's no way that he can patrol the boundaries of this harem. And so they have to be motivated as well to want to interact with him and stay with him. But this isn't all, sunshine and rainbows. Unfortunately, it's also this pair bond, this cross bond is really intensely aggressive. So the male is constantly neck biting and aggressing on females in order to maintain the cohesion of his unit and maintain in both time and space. Okay, so let's get into oxytocin. That's the breakup of the baboons. That's the dynamic. What about oxytocin and vasopressin? Well, they're incredibly useful and interesting compounds, neurohormones. As you can see here, they have an incredibly long phylogenetic history, and their homologs are found everywhere. I mean, even there at the bottom, you can see that they're connected with invertebrates. And their structure has been highly conserved too. Now, typically, in the most simple sense, they're produced in the hypothalamus, specifically the paraventricular and supraoptic nuclei. There's a few more for vasopressin, but for the moment, I'm just going to talk about those two. And it can act via 3 mechanisms. So in one way, oxytocin and vasopressin can be act as hormones, where they're released into the posterior pituitary to go into circulation in the blood. However, I also want to point out that there are recurrent collaterals there. In other words, as the axons that project from the hypothalamus to the pituitary have branches that go backwards, back into the brain to target specific sites. So in that kind of sense, you can have a correlated action where it's being released into the outside and the inside at the same time. It can also act as a neuromodulator, meaning that it's typically, it can be released by the body of the neuron itself and just go into the extracellular space where it can change the general activity and the excitability of nearby neurons. And then lastly, it can act simply as a neurotransmitter, where especially by these parvocellular neurons in the paraventricular nucleus, which have connections throughout the brain, especially to reward areas, to sensory areas, those sort of areas. That's where it can act. Okay, but what do they do? So why are they so relevant to baboons? Well, oxytocin, in its most fundamental way, is anxiolytic. It decreases anxiety. And while its functions are more well known, maternal behavior and allopaternal behavior or alloparental behavior, it's also essential for female pair bonding, female partner pair bonding. And in general, it enhances approach behaviors, partly possibly because of this anxiety reducing effect. So it enhances the conditions that would allow two primates to interact with each other. It decreases anxiety associated with those sorts of events. It also enhances prosociality, especially in humans, as we've looked. It enhances cooperation as well as trust and altruism, although I feel compelled to mention that this is often only within the context of an in-group. So out-groups, it can be reversed sometimes. But in general, we can see that oxytocin can stimulate and is secreted in response to affiliative behaviors, like grooming in primates, which is a highly affiliative behavior. Okay, what about vasopressin though? How is that connected? Well, vasopressin, as kind of an opposite number, is anxiogenic, so it increases anxiety. And while it is essential for paternal behavior and male pair bonding, male partner pair bonding, this is all within a kind of context of possessiveness, partly because it's built out of these territorial, aggressive, and mate guarding mechanisms as well. This is a very important hormone to mediate those sorts of behaviors. However, I have to also point out that it's a lot of times dependent on the receptor distributions for these hormones in the brain. So this is the classic example of the pair bonding versus promiscuous voles, where what you see is that pair bonding species have a greater density of these receptors, both the oxytocin and the vasopressin V1A, in reward areas of the brain that is absent in very similar otherwise, but promiscuous voles. But so how can this help us? So how can we connect these two lines of research together? Well, people have suggested that there are species differences in oxytocin, and that oxytocin values within a species can give you some clue as to their temperament, the general temperament of the species. So a famous example was bonnet macaques, where the study said that the bonnet macaques were less aggressive and more affiliative than the corresponding pigtail macaque. And what they found was that subsequently, bonnet macaques appeared to have much higher concentrations of cerebral spinal fluid oxytocin relative to the pigtail macaque. So the conclusion was the bonnet macaque is more affiliative, possibly because they have higher oxytocin in their systems. What about individual personality? Well, this has also been well looked at. So oxytocin, we know, even within a species, is very related to affiliative behavior. So for example, among rhesus macaques who are raised by their mothers as opposed to nursery, mother-raised rhesus macaques are more affiliative and less aggressive and have a correspondingly higher concentration of CSF oxytocin as well. So again, another evidence for this idea that individual personality traits can be mediated by differences in the concentration of these hormones. Oxytocin has also been found, especially in the cerebral spinal fluid, to be negatively correlated with your lifetime history of aggression and positively correlated with affiliative behaviors, especially in parents. But what about vasopressin? Well, vasopressin, given its role in aggressive things, it's unsurprising to find that in humans, it is in the cerebral spinal fluid positively correlated with a lifetime history of aggression. More interestingly, in recent work, it's also been shown that it's also higher in more sociable rhesus macaques. So in this study by Parker, we see that that increasing CSF vasopressin concentrations are also linked to very specific things like more time spent in grooming behaviors. Okay, so we've established there seems to be a basis for the question here and the way that I'm asking it, but where to collect it from? Well, the most obvious place is to say, I'm going to go straight to the source. I'm going to go right to the brain. I'm going to collect it from the cerebral spinal fluid. The problem, well, the good thing about that is that it's very close to the brain. It can be thought of as an indicator of perhaps the current activity in the production of oxytocin, or even as a pool that can continue to be bioavailable for oxytocin use in the brain or vasopressin's use in the brain. The con, obviously, this is incredibly invasive. It's very, very hard to do, and it's very stressful for the animals as well. So that's a problem. So another option, of course, is to look at peripheral sources like the blood plasma, the urine, or the saliva. These are great for field research. They're much easier to collect. Well, at least urine and saliva. Blood's still a little tricky. But either way, it's great for field research. The problem is, again, it's probably more of an indirect connection to its influence on the brain. And also, we're not sure yet how it's actually influencing central activity. So people tend to use it then as a proxy for central activity. But then that raises the question of, well, what do these individually mean? And more so, how do they relate to each other? So how do these sources act independently with behavior? And how do they act jointly, if they even act jointly at all, in the mediation behavior? And that's why I wanted to bring up again this case of coordinated release of a peripheral into the blood and to a central. So my hypotheses and predictions for this, well, my primary aim was, again, this between subspecies comparison. And very simply, my prediction was they're going to differ. Somehow they're going to differ. My specific hypothesis, which is a weak hypothesis, by the way, was that I believe that Hamadryas baboons would be greater in their oxytocin and vasopressin concentrations, probably sex differentiated, due to their greater reliance on bonding mating relationships. That was my belief at the time. And that specifically, I would see these differences in the cerebral spinal fluid, not in the periphery. And my primary aim too was to look at how do these concentrations align with an individual's typical behavior, their typical social temperament. And what I suggested there was that individuals that have high concentrations of oxytocin are going to be more affiliative in general. Likewise, if you have higher vasopressin, you're going to be more aggressive in general. And that, again, I predicted that this would be in the CSF, not in peripheral sources. So how did I do this? Well, I was very fortunate to be able to find baboons. I had to find them first. And luckily, there are plenty in the United States. There's 300 plus at the Mannheimer Foundation, who I thank you guys very much. I mean, both the Mannheimer Foundation, which has Hamadryas baboons, and Baboon Research Resources at the University of Oklahoma, which has 600 nudus baboons. I cannot thank them enough. They helped me tremendously throughout this project. But yeah, and they both had these large mixed sex and mixed age corrals where the baboons could act quite freely. Now, as for the hormones, what I did here is that I collected hormones, unfortunately, only at one time point. It would have been nicer to get multiple time points, but this was just what the time allowed. So I collected all hormones at one time point from 84 baboons, which were fairly well split between Anubis and Hamadryas, and male and female. And all were adults or just subadults. And from each of these individuals, I tried to collect 2 milliliters of cerebrospinal fluid, 10 milliliters of blood, which was centrifuged down to 5 milliliters of plasma, and then 10 milliliters of urine as well. And all of this I then sent to the Wisconsin National Primate Research Center, where it was assayed using ELISA. Okay, but then how did I actually look at the behavior as well? Well, with the behavioral collection, I had 91 baboons, although I only had hormones for 70. This is also quite well split, 34 and 36. And what I did was I did 20-minute continuous focals, which basically means I would pick a female like the one here, Cinnamon, and say, I'm going to follow Cinnamon for 20 minutes and remark everything that she does to another individual or that they do to her that's within my ethogram, the behaviors I was interested in. And I tried to collect at least 4 hours of data per each individual. I then wrote, as Adrian pointed out, I wrote a very inelegant script that managed to, in R, in order to collate all this data that I transcribed into a usable form as well. So you can see a lot of if and for loops there, which I know are very inefficient, but it worked. Then, but in terms of what behaviors was I actually looking at, or that I'd be looking at in these papers, my response variables were comprised of events and states. So what I mean by an event here is something like approach. You can see right here, one baboon has just approached by walking within 1 meter of another adult. That's an event. They just walked there and they even just sat down. A close approach is the same sort of thing, except they're now going to move to either being in contact or just within 10 centimeters, which is practically in contact. And then grooming is the same idea, is that it's the moment where you initiate grooming. Now, these are also related to different states. So being in proximity is what that baboon is doing right now. It's sitting within 1 meter. So it initiated by approaching, and now it's in the state of being in proximity. And we can then think about, well, how long is it going to stay there for? And how does that relate to hormones? And so you can ask the same thing about how long are they going to sit in close contact? How long are they going to groom each other? And then lastly, I have some composite events, such as dominance and submission. So these are a series of different behaviors like threats or attacks. Like this male Hamadryas baboon right now is ground slapping. That is a very, that's his way of telling you that you're pissing him off. And so I would register that as, for example, as a threat given. And I also looked at self-directed behavior, since that's a really good indicator of how should I put it, of stress and anxiety, since baboons or even primates in general that tend to self-groom or scratch too frequently or more frequently than others are often highly stressed. Now, as for the data analysis, as Adrian alluded to earlier, thanks Jordan and Eric and Adrian, but Jordan and Eric helped me a lot with this. I did a Bayesian analysis, and the reason for that was it was a better fit for my data, for me to ask the question that I wanted to ask, as opposed to having to fit my data around existing kind of tests, like t-tests or whatever. This was a way that I could test my model in one go. Also, it's a great way to impute any missing values. In some cases, I had plasma, but I didn't have CSF for an individual. Well, through this, I'm able to more accurately impute probable values. I used R and BRMS to do this. And for later on, in order for people to understand the interpretation of the data that I'm showing, every parameter will have what's called a posterior distribution. And so I'm not showing p-values, I'm showing pp values, which are a proportion of the posterior that supports a conclusion that I'm drawing. So if 95% of a posterior of one parameter is completely different from another, then we can say those are separate. They're not the same thing. Same thing, we do 95% credible intervals, which is saying 95% of all posterior samples are located within this region. All right, so what did I find? Study one with the between subspecies comparison. Now, the model for this is clearly straightforward, so I don't think I need to explain that too much, this block of mathematical text. But what it is it's a multi-level and multi-response model. The multi-response is referring to the fact that I have 4 hormone response values that I'm looking at: the CSF oxytocin, plasma oxytocin, the urinary oxytocin, and the CSF vasopressin. All these were log transformed and centered, and the urinary was additionally corrected by creatinine levels, since urinary oxytocin can be affected by dehydration. And the great thing about using them as a multi-response model is that I can then say, once I've accounted for all these other parameters, then I can say, well, what correlation is still left between these different responses? And that's how I can get at this idea of the source correlations between CSF, plasma, and urine. I also included corral ID as a random effect. Potentially being a baboon in corral 3 is not the same as being a baboon in corral 2. So that has to be accounted for. I then had a number of fixed effects, which I included also subspecies, sex, and age as a 3-way interaction, partly because earlier research in baboons had suggested that there might be age and sex-related differences, and then it all kind of worked together. I also included a spline for data collection, for spline fans out there. As for subspecies differences, so this is what you came for. This is the big one. What subspecies differences did I find? Big fat goose egg. No subspecies differences at all. They're very close to one another. Yeah, there we go. The Price is Right theme. I don't know how many of you even watch Price is Right. I don't know, Adrian, you're in Switzerland. I don't know if you even got that, but but yeah, so no subspecies difference at all, which is fascinating. Why? Why is there no difference? But there were sex differences. Interestingly enough, what we found was that cerebral spinal fluid and urinary oxytocin were greater. It's a strong effect. It's not a super strong effect, but it's still an effect. In the CSF and the urinary oxytocin were much higher in females than in males. There was a weaker effect much weaker effect for the plasma oxytocin though. So in general, oxytocin seemed to be higher in females than in males. This is interesting too once you add in reproductive state. So if you are cycling, if you're lactating, or if you're pregnant, this also had a very strong effect. Where interestingly enough, the cerebral spinal fluid is almost entirely unaffected by your reproductive state. It seems to be quite insulated from it. Yet the periphery is highly influenced by it, but specifically not by lactation, only by pregnancy. And it occurs in the plasma and the urinary, where it's much, much higher in pregnant girls. And then unsurprisingly, considering this importance of pregnancy, it's also a huge effect on gestational day as well, where thankfully the people at Mannheimer, when they were collecting hormone data from the Hamadryas baboons, they were even able to get a good estimate of likely gestational day of some females, which allowed me to actually ask, well, does it change throughout gestation? And sure enough, the closer you get to birth, the far higher your values of oxytocin in the plasma and the urine. Again, it's unaffected in the cerebral spinal fluid. And then lastly, of the parameters that I was interested in too, I also had age. There's a lot of reviews out there on oxytocin and age, and they all say, wouldn't it be great if we knew more about how oxytocin changes with age? And so this is kind of a cool way to show a little bit more of what happens here, where what I found was that in this case, oxytocin interestingly decreased in its concentration with age in the CSF and in the plasma, slightly weaker in the plasma, but it's still decreasing in the plasma as well. While paradoxically, it's increasing very highly in the urine. And this is something that's kind of confusing because we thought at first maybe it's due to creatinine that value is changing, but it didn't seem like creatinine was changing. So we have a very weird kind of pattern there. And as for the residual correlations, again, what correlations exist between the sources in baseline hormones? Another big nothing. Very interestingly, there's no correlations. The strongest correlation that I found was between urinary oxytocin and CSF vasopressin. And as you can see from that, it's so uncertain. That distribution is very wide and flat. And so it makes it a lot harder to say for sure. Is that really a real thing there, or is that there's just too much noise? Okay, so going into the discussion of this first paper, no subspecies differences, which I think is a really fascinating result in its own way. I would have preferred that there were, but well, take what you can get. So the reason why, well, maybe there's developmental differences. This is one thing they brought up in the case of the baboons that Clifford Jolly studied back in 2008. What he found was that there were differences between Hamadryas and Anubis males in relation to the CSF monoamine metabolite for serotonin. And he found it in relation to age, and especially age at dispersal for Anubis males and age at harem formation for Hamadryas males. So what he found there was specifically where that I just circled is there's a huge dip in Hamadryas male serotonin levels at that age where they're forming harems, because the idea is that possibly they need to be more impulsive, they need to be more aggressive. And that's one thing that having lower serotonin levels typically leads to. While the Anubis baboons have much higher levels of serotonin, because as they disperse, they need a lot more social savvy in order to get into a new group. Meanwhile, then, as you go through life, the need of maintaining a harem is entirely different, or the skills you need to maintain your harem is entirely different than the skills you needed to acquire your harem. And so in that case, you see that it kind of goes up a little bit higher at that point. So the serotonin levels change. So maybe there's a developmental aspect. Also with the bonnet and pigtail macaques, that study which showed the differences, that could be also related to the fact that they had very biased sex samples. The more affiliative species, the bonnet macaque, their sample, when you look at it, is mostly females. And as we found from our own study, females tend to have higher oxytocin than males. So it's highly possible that the result they found is confounded with the fact that they had a biased sex sample. And also, it could just mean that within subspecies variation is far greater than between subspecies variation. And although I would put my money on the last one, which is receptor distributions. And I'll talk about that a little bit later. Also, sex differences in oxytocin were not terribly surprising. There's been a meta-analysis that's looked at that too and has found a similar result. Also, we know OT secretion is affected by estrogen itself and estrogen levels. As for the reproductive state differences, this is also something that's been observed before. So CSF is generally unaffected by whatever pregnant or lactating state you're in. Rather, it's the periphery that is far more affected by it, but specifically by pregnancy and not lactation. This is actually a study here by Amico, where the black bar regions represents where the mother is actively suckling. And what you see is that those active suckle areas lead to an increase in plasma oxytocin, but it returns to baseline as soon as they stop suckling. So it actually goes down. So you wouldn't expect to see any differences if you're lactating, at least in baseline hormones. And lastly is age. There's actually been a few other papers that have also found a decrease in CSF oxytocin with age, like this Parker paper in rhesus macaques. But it raises an interesting question about why. Why is OT decreasing with age? I read, I believe it's Volstad's review recently where he looked at several demographic variables in oxytocin. And at the time, he said that he found that in many studies, oxytocin appears to increase with age. And he had an explanation for it. He's like, oh, well, that makes sense because you're doing this and you're more sociable in that. And I'm finding the exact opposite. And although I do find what he found, but in the urine, which might have caused him some issues perhaps. So it's an interesting question about, well, why is it even changing? What does this relate to? And what are the behavioral consequences of these changes? And then lastly, the residual correlations. Baseline oxytocin and vasopressin sources were uncorrelated. This is, in particular, the CSF and the plasma was unrelated in these baseline kind of situations, even with pregnancy, considering how much of an effect pregnancy had on the periphery. It had no impact on the central levels. And even plasma and urine were unrelated, although that's more likely due to a time delay in the way that urine, oxytocin reaches the urine and then excreted. So the answer is likely going to be a contextual one. So this paper right here, this is the Volstad meta-analysis I was talking about. And what he found was correlations between at baseline, correlation between sources at baseline almost are zero all the time. They're never really correlated. Rather, you see correlations between sources in very specific scenarios. So like he used the example of the stress. So under stress, you might see correlated release of oxytocin both centrally and peripherally. So looking future at what exactly is the correlation, I think we can say that baseline hormones are uncorrelated generally, as far as I can see, but that we have to look at specific behaviors to see how those get correlated with each other at that moment in time. Now, as for the between individual comparison, compared with personality, the model here is very similar, except now I have 9 behavior responsive variables that I mentioned earlier: approach, close approach, groom, etc. And this time I also included 3 random effects. I included not only corral, but also subject ID and observation ID, because much the same way that a baboon from corral 3 could be different from a baboon from corral 2, every individual can be very different in their propensity to certain behaviors, and even a specific day that they had an off day. they had a day where they were not as affiliative as they normally are. So it's important to keep these things accounted for. As for the fixed effect parameters, I had my 4 main real experimental parameters were looking at the hormones. I wanted to know, how are these hormones affecting the expression of affiliative and aggressive behavior. I also included, though, control parameters such as sex, in case that might have had some sort of effect on the data, so that could be accounted for. What I found here is far more fascinating. So with 1 meter proximity, that's just approach and being in proximity, that was had very weak results in general. You didn't really see strong associations with 1 meter proximity. The CSF oxytocin does have weak associations, like here and here. And actually, if you look at it on these graphs as well, showing the marginal effects, what you see is that even though it's weak, individuals with high CSF oxytocin tend to approach more frequently, and they tend to spend more time in proximity. So that's at least a tantalizing suggestion. However, it's much stronger when you look at close proximity behaviors. So in this case, when you're sitting within 10 centimeters, or you've approached within 10 centimeters or in contact, you have very strong effects, and specifically in the plasma. So plasma has a very strong positive relationship, while CSF has a very negative relationship to the state of being in close proximity. So the way to show it kind of here is that you can see that as you increase, or individuals with higher plasma oxytocin tend to approach in close far more frequently than ones with lower. And furthermore, baboons, yeah, that's what happens when you study them for too long, you call them people. Baboons that have higher levels of CSF vasopressin tend to spend less time sitting in close contact with another individual. And then this is repeated again in the grooming behaviors with a slight twist. And grooming behaviors are also very strong again in the blood plasma, where plasma has a very strong positive relationship with initiating grooming. And this time CSF vasopressin has a very weak association, but positive one, with the length of a grooming bout. And so what we're kind of saying here is that with individuals with higher plasma oxytocin tend to initiate grooming more often. So like before, they initiate close contact more often, they initiate grooming more often if you have higher baseline plasma oxytocin. And as I said, interestingly, individuals with higher vasopressin, they actually tend to have longer grooming bouts too, apparently, or spend more time grooming. As for aggression and submission behaviors, this was also fairly interesting and a little confusing. What I found here was that urinary oxytocin, it was found to be weakly associated with aggression. So if you had higher urinary oxytocin, you would produce fewer aggressive behaviors, which thankfully makes sense with the opposite, more stronger finding that if you have of a positive effect with submissive behavior. So if you have very high urinary oxytocin, you tend to produce more submissive-like behaviors. Plasma oxytocin, though, on the other hand, has a completely opposite relationship, where it has very strong negative association with submission. In other words, I'm just going to show right here, just the submission data, is that with plasma OT, the higher your plasma OT, the fewer submission behaviors you produce or that you act. While with urinary OT, it's the exact opposite. The higher your urinary oxytocin, the individuals with that tend to produce more submissive behaviors. Okay, so what does that mean? So how do we make sense of these results? Well, the most interesting thing is that, hey, the hypothesis was somewhat supported, this idea that there are associations between affiliative temperament, affiliative behavior, and your concentrations of oxytocin. So specifically, initiating close contact and initiating grooming are very strongly related to your concentration of oxytocin. And the important thing to consider here is that these are both highly intimate acts and they're incredibly dangerous acts too, in a sense. I mean, if you think about any one of us right now, if you're just walking a subway and just sit down next to somebody when the whole bench is open and just sit down and start touching them, you might get attacked. Similar in the case of baboons. They don't like it if you just show up and start touching them. And so there's a high possibility that if you just go and approach somebody in close and try to initiate grooming, they can attack you or they can reject you in some sort of way. So, but how is OT doing this? How is OT increasing these initiation behaviors? Well, the way I see it, it could be a combination of two things. It's affecting your social perception and it's affecting the internal state. What I mean by this is that it's blunting negative aspects or negative perceptions of the partners that you're engaging with and the potential outcomes of these engagements. And it's also just in general decreasing the internal anxiety of trait anxiety of the individual, which allows them to even initiate these things in the first place. Likewise, that decrease in baseline anxiety also increases baseline trust as well too, which is an important aspect, that you're going to trust whether or not it's a good idea to walk up close to this other baboon. And what I mean about these kind of perception and internal states, especially the perception, is shown in what we've already found in a lot of primates. So this great study by Ibbotts, where he found that among rhesus macaques, if you give them oxytocin, what tends to happen is that they spend less time looking at high-ranking faces and they're less distracted by threatening faces. They're less distracted by fearful faces. They're less distracted and afraid of dominance faces. There's a paper by Zhang that came out in 2018 that referred to this as flattening the dominance hierarchy, which is a phrase that I really like. It's this idea that oxytocin has this ability to make dominance relationships seem less severe, which is an important part if you want to interact with somebody who's a higher rank than you. Even in our own department, Jim Rilling has looked at cooperative effects of oxytocin. So you see, in this case, in the prisoner's dilemma, that if you give a male intranasal oxytocin, they tend to cooperate even after their partner has defected. So they're not thinking about the consequences of even the action. This is the same as a Baumgartner study, which showed that even when your trust is betrayed, people who are given intranasal oxytocin continue to trust. So it's definitely changing how you perceive your partners in an interaction. But why is it only for events? That was my question. Why isn't it affecting the time grooming or the time spent in close contact? Why is it specifically for events? And I was going to suggest that perhaps it has something to do with that oxytocin is reducing the anxiety associated with that acts as a barrier to simply initiating a social act. Like a social act itself, the decision to act socially is a hard decision to make. And what oxytocin does is it decreases the probability that you'll retreat, and more so increase the probability that you're willing to approach and engage in the activity. And after that, it's other mechanisms that say how long you want to stick around for. But then the more interesting thing is, well, why is it in the blood plasma? I would have expected, most of us would have expected, it would have been in the CSF. It's closer to the brain. It's more likely to affect behavior in there. Why is this happening in the blood plasma? And it's not the first time this has been observed. I mean, Mastropieri in 2009 and Berg here showed that macaque typical behaviors, like in this case, maternal behaviors and prosocial behaviors, are related to their baseline levels, the baseline plasma levels of oxytocin. So it's not a new finding, but why? And so my thought about plasma OT is, if in fact this is related to anxiety and reducing anxiety, that there's a couple of factors that are important. Number one, that oxytocin is strongly related to the functioning of the adrenal glands. We know hypothalamically it can decrease the production of ACTH. And there's also evidence to suggest that it's able to decrease the production of cortisol directly as well. There was this great paper by Legros in 1988, which showed that if you gave two groups of people an injection of ACTH, which is a precursor to cortisol that needs to be converted to cortisol in the adrenal glands, if you gave them an injection of it, it led, of course, to an increase in cortisol. But if you also gave some of them an injection peripherally of oxytocin, it led to a substantial decrease in the amount of cortisol that's produced. So it's suggesting that perhaps somehow plasma oxytocin has an ability to decrease even the production of those cortisol signals to the brain that you're stressed. On top of that, we know that OT receptors are everywhere in the viscera. They're all over the place, especially like the adrenal glands, but also the cardiovascular system. And so that's another way that they can act in a bottom-up sort of way to behavior, by adjusting the body's current state to say that it's more relaxed, like adjusting heart rate or adjusting the dilation of arteries. So those are different means by which oxytocin could be, I don't want to say fooling the body, but in a way it kind of is fooling the body that things are fine. It's okay. You go walk up to that giant male and just start tapping him on the back. It's okay. That's a little extreme, but it gets the point across. And then lastly, another option too is that it's communicating via the vagus nerve. The vagus nerve is a very important cranial nerve. It's the way by which all of the internal signals are being shuttled to the brain about internal state. And people have always said, well, maybe OT is working through vagus. Maybe OT is working through vagus. And there was actually a study that came out last year, 2019, which showed that this could be true. So what they did was with these rodents, they injected them with peripheral oxytocin, or they injected them peripherally with oxytocin. And what they found was on this y-axis, it says CFOS, immunoreactive neurons, in the PVN. What that means is that this, it's a measure of kind of like the activity in that area. And what they found was that when you give them this blood infusion of oxytocin, it actually increases the activity in these central brain hypothalamic regions. And interestingly, you could abolish this effect by simply just giving a vagotomy and cutting the vagus nerve. So it's suggesting here that, again, OT could be communicating in a bottom-up sort of way, the central behavior, via changing the reactivity of the vagus nerve, or somehow influencing the vagus nerve's activities. AVP is a bit more confusing. I mean, you can make stories for all sorts of them. I mean, you have individuals with high AVP have shorter time in close contact. Maybe it's because they're so stressed that it's harder to sit a long time. It just sounds like a just so story. But at the same time, we also see that individuals with high vasopressin spent longer time grooming, spent more time grooming. And I find that very interesting because that does correspond to the recent paper with Parker, which found a very similar finding, that increases in CSF AVP concentration led to something very specific, like increasing time grooming. And maybe it has something to do with sex differences. I believe the Parker paper was in males. And from what we know from Dr. Rilling's work, Jim's work, is that there are sex differences in how the different sexes react to differing values of these hormones. So the way that the males reacted to a cooperation test was not the same way that females given these same hormones reacted to a cooperation test. So maybe it could be something like that as well. And lastly, the dominance and submission behaviors. This was also very confusing. The way that I interpreted it is plasma oxytocin was negatively associated with performing submissive acts. And what we know from humans and other primates is that people who have suffered abuse in their life have lower values of CSF oxytocin. Also, people that have high anxiety or depression can have elevated levels of oxytocin. And I consider that in some cases to be very similar to what it's like to be a low-ranking baboon. That's one way to think about it, is that these ones they're abused to some extent, and they're constantly, in some cases, they're without support from family members, which is where you'd normally get some of that kind of buffering. So perhaps what's going on is that these individuals that are low ranking have incurred a lot of these problems and are highly stressed out, and that's why they have lower OT values. As for urinary oxytocin, that's even more confusing. The only thing I found previously was Feldman's papers showing that baseline urinary oxytocin is positively correlated with relationship anxiety and relationship stress. And another study that said that baseline urinary oxytocin is positively correlated with the expression of psychopathic traits. So it's a much harder thing for me to unpack what exactly is going on with urinary oxytocin. Urinary oxytocin in general seems to be very interesting and very different when you're looking at it baseline relative to if you've collected it just after a social interaction. All right, so just to wrap this up, conclusions in my future directions. Well, the main conclusion is they're not different in their OT and AVP concentrations. And I believe that the answer is going to be, if there is any relationship between the OT and AVP systems between these baboon subspecies, it's going to probably be in the brain. And I've already begun working on that. As you guys can see, I have some beautiful baboon brain pictures here of autoradiography for the V1A vasopressin receptor. So those are some baboons right there showing the distribution. So keep your eyes out in the world for that paper to come out. And then also, the other big conclusion is that individual personality and temperament are definitely represented, even in baboons, with oxytocin and vasopressin concentrations, particularly in the blood, interestingly. But what else could be related? So I'm interested too in saying, well, what about, these are common questions among primatologists that I think could be applied here, which is, well, what about relationship symmetry? So how often individuals that groom as much as they get, how about them? How does that affect it? Could that be a connection versus ones who are very asymmetrical in their relationships? Likely, again, to developmental. Again, this has been an aspect that I think is really fascinating, is the idea of, especially when you're going through big life changes like dispersal in primates, excuse me, what is going on in these systems at that point to prepare them for these large changes in behavior? And I think we just haven't really looked at that yet. And lastly, I just wanted to even bring up something I haven't talked about, which is genetics. We've actually already, there's a paper that came out by Christina Berge just a few years ago about baboons and how they differ in their dopamine receptor pathways, or excuse me, dopamine, yeah, I believe dopamine receptor pathways, and how that could be another way in which we can get at this question by looking at the receptor genetics of the two baboon subspecies and comparing and see how that relates as well. So I just want to thank everybody who came out here today, especially my wife, my family, and all my friends, my committee, both past and present. I want to thank, of course, Adrian, who's helped me so much, Dr. Preuss, Dr. Young, Dr. Stout, Dr. Rilling, and Dr. Wilson, and Dr. Hooper. I forgot to put his name in here because I haven't seen him in a while. And I promise I will get all of my junk out of your fridges, by which I mean oodles and oodles of baboon urine. I will get that all out, I promise. I also want to thank Christina Rogers and Eric Ringring and Jordan Martin, who have helped me immensely, especially Christina, who We spent many hours till 3 in the morning cutting brains. I also wanted to thank everybody at the baboon colonies that I worked at, especially doctors Pablo Morales and Vito Palmieri and Dr. White as well. They were unbelievably helpful in all aspects of my work. I want to thank my old cohort. Sorry, it took me so long to finish. And the whole Emory Anthropology Department, especially Laura McDonald, who has been helping me, dragging me to this finish line, I could say. I'd also personally like to thank the Young Lab and the Preuss Lab. Thanks, Marianne, who's been so helpful, for being so helpful. Everybody in those labs really accepted me as one of their own, and I appreciated that a lot because it helped a lot. I also want to thank Dr. Lauren Post, and of course my funding sources, the National Science Foundation and the Silvio Oconti Center for Oxytocin and Social Cognition, which gave me the opportunity to work on these sort of projects. So thanks everybody. I see a lot of clapping and I hear spattering of clapping, so that's good.
So I think in general with this number of people, it usually seems to work okay. If people just unmute themselves to ask a question, we can, if we start getting crosstalk, we can start using the chat for people to kind of queue up. But at least initially, I would say, if there's a silence, feel free to speak into it. With the proviso, they'd like to start with the with graduate student questions.
Don't all talk at once.
I can go. Hey, Dan.
Hey, Eric, how's it going?
Good. Hey, congrats and great job.
Thanks.
So I had a question. So you have these big gestation effects on the hormones.
Mm-hmm.
And I was wondering how that played out in the behavior too, because like,
That's a good point.
Are there any sort of unique behaviors that you would say either within the ones you measured that were different among the pregnant baboons, or maybe unique behaviors that you saw exhibited by the pregnant baboons?
So it's actually great you say that. You've pretty much summed up all of my fears throughout July and June. I was like, oh God, how are these going to screw things up? Because I was worried about that exact question. Because although I don't know if I necessarily saw myself a lot of behavioral variation based on their pregnancy or their lactating status, I know other people have. Other people have mentioned, especially mostly in the case of Hamadryas baboons, that they change their general distance to the OMU leader based on who's probably pregnant. But I also think that's in wild circumstances where often there's just two females that are with the male, and so they're kind of shifting off with each other. And so it was kind of hard for me to say. At least when I look at that control parameter and how it kind of plays out in terms of its effect, it didn't have any real huge effects. So like just on behavior, I didn't find it. It caused me a lot of problems too, because it was highly variable. I mean, it had an incredibly flat distribution. It didn't really have any kind of thing, but it was all centered at 0. So it doesn't seem in this case that it had a huge effect on the behavior. But like I said, I have heard of that occurring in the past.
Yeah, because you wonder sometimes with behavioral biology, sometimes like with oxytocin especially, pregnancy is almost like an alternative behavior. It's like, oh, maybe it's for behavior or it's just for pregnancy, but why not both?
Yeah, exactly. Yeah, no, I really don't know. But yeah, like I said, I honestly think one of the things that would have been perhaps more important that I got, but I didn't really have great data on, I didn't keep great records of it, was position and cycle. So whether or not you were at that moment in estrus or anestrus, or what was the level of tumescence of your sexual swelling. Right. I think that might have been more informative in the long run to have known that information than even the reproductive state, like pregnant or things. I think those might have a larger effect, because I did notice those ones. I noticed more clearly behavioral differences. So females that were, for example, in estrus, even among the Hamadryas baboons, acted very differently than if they were anestrus at the time.
Gotcha. Thanks.
Yeah, no problem. Thanks for the softball question. I called him up yesterday. I'm like, hey, you got to give me easy ones, man.
Hey, Dan, I'll follow up Eric's question. So,
oh man, Jordan, how's it going? I'm glad to see that you listened to my my whole defense outside in the beautiful Zurich air.
yeah, I'm getting a great breeze up here. The Wi-Fi is kind of bad, but but yeah, great job. I really liked your presentation. So I was interested to think about the kind of points you mentioned at the end for future directions about distribution of receptors versus baseline levels. And so maybe thinking about how your study informs bigger questions about human sociality, or let's just say sociality in general. Like, do you think that there is going to be an answer? And if so, where does the evidence point about the relative importance of one or the other for explaining individual differences? So maybe to frame that more directly, like, do you think that baseline levels are in general going to be a good predictor of consistent individual variation? Or do you think that receptor density is more important? Or do you think it's just going to vary by species and it's not going to be a predictable pattern?
That's a really good question. That's a really, really good one. Now, I'm not, so that's actually, the hardest thing about that is I don't know how fine grained we've gotten in humans, at least in terms of how do receptor distributions vary with, say, mating behavior. I know Hasse Wallam has done, I know he came to, before he came to Emory, he had done this great thing about looking at marriage, I believe, and divorce related to your vasopressin receptor gene. And he found some interactions there. I know in humans, we found, like Sarah Freeman looked at, for example, autistic people versus non-autistic people, or I should say autistic children and non-autistic children, in terms of the distribution and density of receptors, oxytocin receptors in their brains, and there was a difference. It's not as dramatic as you would see in, say, the voles, but there are differences. Even among voles, actually, within prairie voles, there's a lot of difference in their behavior and a corresponding, I think, variation in their density of their receptors. So I think that's actually a question that I would love to see answered. I think I would love to see more about collection of more like personal variables. So like, how many marriages did this person have? How happy were they in relationships? And then connect that with actual brain data as well. I think that's a far way off, but I think that's a very cool thing to ask. Maybe the genetics might be a better way to go with that. As for the concentrations of the hormones, those also have an effect. I mean, like I said, going back to the autistic, non-autistic divide, I mean, they have, I know Elsa Andari, who was here at Emory, she did a lot of work with this, and she found that I believe autistic children had lower baseline plasma oxytocin than non-autistic children. And we know from intranasal studies that it seems to help them by giving them this exogenous source of oxytocin. So I don't know which one's going to be more important in the end. I think it's because in humans too, I just don't know the dynamics about the release of oxytocin in relation to a social event. You know what I mean? So like if you and I started grooming right now, I don't know how that would relate to my increase in oxytocin secretion rate or something like that.
I mean, mine's going way up. But
oh, I knew it. Just it's like it's like those studies of humans and dogs where when they look at each other, their oxytocin increases.
right.
Just like looking at each other. But yeah, no, that's a really good, complicated question. That's like a Christmas kind of like wish question, where it's like, if I could wish for one thing, I would love to have as many human brains and all the background data on them that I could get, and look to see how that correlates to differences in these kind of things.
Cool.
That's potentially something I could do with baboons in the future too, if you just keep collecting males, because all the males that I worked with differ greatly in their bonding behavior. I mean, some males had very large harems, others had very small in these conditions. And so that could even be looked at on a finer level.
I don't want to steal any more time, but just to make sure I